Hello and welcome. Today let's continue through with our nervous system and let's finish this part off. So here today what we'll do is we're going to now pick up with the synapse. We have gone through and we've taken care of the dendrites. We spoke everything about the dendrites, how they're the incoming receptive region, and the signals that they receive are going to be graded potentials. And the cell body we saw also receives information, and then it's got other functions, produces neurotransmitters, proteins, and whatever else it has to do there to maintain the neuron. And then the cell body is also, the soma is also going to serve as an incoming receptive region. Okay, so then we moved to the axon. We talked about the axon. So graded potentials also can be received on the cell body. Okay, then we talked axon. Axon we talked about and we saw action potentials there now. Totally different signals. If this happens to be your essay, you have to be able to go back to the dendrites and start talking from the dendrites and explain everything about the dendrites and move to the cell body and down to the axon and talk about propagation and talk about how the signal is going to be propagated along the axon all the way down to the axon terminals, where is kind of where we left off, and now this is where we're going to kind of pick up from is the terminals and then move down into the synapse and then move to the next cell and kind of see how this information is going to be processed. Because ultimately, right, we know that the nervous system, as we see here, works because information flows from neuron to neuron or from neuron to effector cells. So when we go through and we talk about synapses, I would like you to know a synapse is going to be a junction. When we talk about a junction is where two things come together. So it's a junction that's going to mediate information transfer from one cell, from you can say one neuron, to the next. So when we define a synapse is defined as a junction that mediates information transfer from one neuron to the next, or from one neuron to the next effector cell, which can be muscle cells. Now, when we talk about a junction you can see is where two things come together. So again, here, information transfer from one neuron to the next. Okay, so the synapse is the junction. That synapse is what we're going to be talking about. Okay, so we're going to see information being transferred from one cell to another. So where two things come together, so one neuron and another neuron, or we said one neuron and an effector cell. We can have synapses between axon endings of one neuron and the dendrites of the other neuron, where then we have what we call axodendritic endings. So again, they're going to be synapses between the axon terminal of one neuron and the dendrite of the next neuron, of another neuron. Now, those synapses between axon endings of one neuron and the cell bodies, the soma, right? The cell body is also known as the soma of another neuron, are going to be called, as we can see here, axosomatic. Axosomatic. So between the axon terminals of one neuron and the soma of the other neuron. Now we can see we have less common types, axoaxonic, axon to axon. Second, we can see here dendrodendritic, dendrite to dendrite. Somatodendritic, the third type, dendrite to soma. So here we can see an image that will kind of depict those two. Axosomatic, axodendritic, axoaxonic. We can see three different synapses here. Now, when we go through and we talk, let's talk some terminology. I want you to know the neuron conducting impulses. The neuron conducting impulses towards the synapse. The neuron conducting impulses towards the synapse is going to be called the presynaptic neuron. It will be called the presynaptic neuron. And the neuron transmitting the electrical signal away from the synapse is going to be called the postsynaptic neuron. The postsynaptic neuron. The presynaptic neuron sends the information, and the postsynaptic neuron receives the information. The presynaptic neuron sends the information, and the postsynaptic neuron receives the information. There are two varieties of synapses. We can have electrical synapses and we can have chemical synapses. What we're going to do here is we're going to look at chemical synapses. We're going to look at chemical synapses. So let's talk chemical synapses then in greater detail. Now, when we talk about chemical synapses, I'd like you to know chemical synapses are going to be synapses that are specialized to allow the release of chemical neurotransmitters. They are specialized for the release and the reception of chemical neurotransmitters. Chemical synapses are made up of two parts. They're made up of two parts. The first part we'll see is the axon terminal of the presynaptic neuron. So first we have the axon terminal of the presynaptic neuron, which we'll see is going to contain many tiny membranous bound sacs called synaptic vesicles. Each synaptic vesicle we'll see is going to contain a neurotransmitter molecule when we look at that in greater detail. Second then, so the second component of the chemical synapse will be a neurotransmitter receptor region on the postsynaptic neuron's membrane. And it's usually located on a dendrite or the cell body, as we have discussed. Okay, those are going to be the two more common ones. So usually on the dendrite or the cell body. And those are the ones that we're going to look at and we're going to talk about. The presynaptic and the postsynaptic neurons may look like they are attached to one another, as we can see here. They may look like they're attached, but they are not. They are not. They are going to be separated by what's known as the synaptic cleft. So here we can go through and we'll talk about the synaptic cleft. The synaptic cleft is a very small, fluid-filled space that's going to be found in between the two neurons. The transmission of nerve impulses along an axon and across electrical synapses is purely an electrical event. However, chemical synapses, they convert electrical signals to chemical signals with the use of neurotransmitters. These neurotransmitters, what they do is they travel across the synapse. They travel across the synapse to the postsynaptic cell, where they are going to be converted back to then an electrical signal. So you can see how that signal can go from being electrical to chemical and then back to being electrical, right in between a neuron and another cell. So here we can see that as well. The transmission across the synaptic cleft is going to be a chemical event as opposed to an electrical one, which is going to be happening across that presynaptic neuron. That'll be all an electrical event, right? That action potential. And then we're going to have the chemical event take place, we said. And then the next cell is going to be able to generate a new action potential. So it'll have an action potential taking place, an electrical event again generated there. Now, this whole event is all going to depend on the release, the diffusion, and the receptor binding of the neurotransmitter. So if the neurotransmitter doesn't get released, this is not going to happen. If the neurotransmitter cannot diffuse across that synaptic cleft, nothing is going to happen. It's not going to be able to bind to the receptor. It's not going to be able to generate anything, right? And third, if you can't have receptor binding of the neurotransmitter, there's not going to be then any type of an event being generated for the next cell, which we're going to see also can happen in muscles. That's where it could become a problem if you're not going to be able to have the receptor bind. You have a condition we call myasthenia gravis, where receptor binding isn't going to be sufficient. And then last, transmission across the synaptic cleft ensures unidirectional communication between the neurons. So let's move forward then, and let's talk about then information transfer across chemical synapses. Here we can see it as bullet points, but we'll go through and we'll explain each of these processes and we'll see what's happening in each one, which is how you would have to do for your essay. Explain everything thoroughly as if you're explaining this to somebody who has no idea. You can even practice with your family members and tell them, this is your ultimate goal. You're going to try to explain to them how information is transferred from one cell to another. And you start by talking about all the events in the beginning, and then you move from the dendrite to the cell body, and you talk about everything that's happening there, and all the events that are happening in the cell body, then all the events in relation to the action potential, how gates are opening and which ones are opening and which ones are closing, and and explain all the way to the end. So here we can see the action potential arrives at the presynaptic axon terminal. And when this action potential arrives at this presynaptic axon terminal, this is going to begin neurotransmission. And then second, we can see here, there's voltage-gated calcium channels here you're going to have to know about as well. I'm going to show you. So you have to be able to talk about those, and you have to be able to talk about what's happening there, and all of that in relation to this neuron sending signals to the next cell. So the second major event then is that the voltage-gated calcium channels are going to open. And when these voltage-gated calcium channels open, calcium is going to enter into the axon terminal. It will enter into the axon terminal. Don't worry, we're going to see pictures that are going to show you all of this as well. Okay, I want you to just kind of learn this by kind of hearing what's happening, and then we're going to put pictures with all of this, and you'll be able to see it all actually happening also. So next then we're going to see calcium's entry is going to cause synaptic vesicles to eventually release neurotransmitters by this process, exocytosis. Now, the surge of the calcium acts as an intracellular messenger. The calcium, it binds with this protein called synaptotagmin inside the terminals. As a result, synaptic vesicles fuse with the axon's internal membrane and then empty their contents by exocytosis into the synaptic cleft. Then we'll see this calcium that rushed into the cell. This calcium is going to be removed. Active calcium pumps will remove this calcium from the cell and taking it out of the cell. Next, then, we can see the neurotransmitter is going to diffuse across the synaptic cleft, and it's going to bind to specific receptors that are going to be on the postsynaptic membrane. And then binding of the neurotransmitter is going to open ion channels. So when this neurotransmitter goes and binds to the receptors on the postsynaptic membrane, what that does is it opens up ion channels. And when these ion channels open up, what this does is, if we're talking about, let's say, another neuron, let's say this postsynaptic membrane I mentioned is a neuron. So on the postsynaptic neuron, it will open up ion channels. Now, when these ion channels open up, you've seen what this does. It creates graded potentials. Now, depending on the neurotransmitter, the postsynaptic neuron may either be excited or it can be inhibited. So the last part talks about how the neurotransmitter's effects are terminated. Now, I want you to know, as long as the neurotransmitter is bound to a postsynaptic receptor, it continues to affect membrane permeability. Neurotransmitters' effects are going to be terminated in one of three different ways. It can happen one of the three different ways. We can have first, you can see reuptake take place. When we talk reuptake, this will happen thanks to either astrocytes or the presynaptic terminal, that axon terminal, the presynaptic terminal. It has sometimes receptors on it that will then reuptake different components. Number two, second, then we can have degradation occur by enzymes. Now, enzymes can come in and take these neurotransmitters from the postsynaptic membrane, or they can take these neurotransmitters that are present in the synapse. And then third, we can have diffusion occur. Diffusion away from the synaptic cleft, where we can have the neurotransmitters actually diffuse away from the synaptic cleft. So these are three ways we can have a neurotransmitter's effects be terminated. Next, then, let's check all this stuff out. Let's see it actually occur. So let's look at these pictures here. So here we could see we've got one neuron here, and we have another neuron right inside of here. This neuron we see right inside of here, this specifically speaking is going to be that neuron's axon terminal. It will be one of that neuron's axon terminals. So this is the presynaptic neuron. This presynaptic neuron, now you can see, is not touching the next cell. There is a synaptic cleft here. It's a potential space we described. Next in here you can see the postsynaptic neuron's membrane. And this membrane you can see has receptors on it that we were talking about. And then here we can see we've got receptors on the presynaptic neuron also. And then here we've got neurotransmitters, chemical signals inside of them. And then here we can appreciate some mitochondria also. So here we can see then the action potential we said is going to arrive down the axon, and it'll make its way down the axon terminals. So here we're looking at one of those terminals. The action potential, now you can see it makes its way down this axon terminal, so it arrives on the axon terminal, and here what that does, then it causes this voltage to change. When the voltage changes, this causes, we said, our voltage-gated calcium channels to open. And when our voltage-gated calcium channels open, this allows calcium to make its way into the axon terminals we described. Now these calcium ions are going to make their way in, and they're going to bind, we said, to protein, synaptotagmin. Now when they bind to synaptotagmin, this is going to promote the exocytosis of these neurotransmitter-filled vesicles. So they're going to, we said, start moving towards the membrane. So here then, calcium's entry causes, we said, the synaptic vesicles to release the neurotransmitters by exocytosis. So here those neurotransmitters are being released. Now when these neurotransmitters are released, we said they have to be able to diffuse across the cleft and then bind to the receptor as well. So here we can see they diffuse across the synaptic cleft, and they go and they bind to specific receptors. Will all of these receptors be susceptible to these neurotransmitters? We don't know that. Some may not be. So here then these neurotransmitters are going to bind to specific receptors on the postsynaptic membrane. And when they bind to those receptors, we said they're going to allow ions to move. And when ions move, this generates graded potentials. Now we can have either reuptake take place, we said. So you have to be able to describe all of this in the essay also. So you have either reuptake occur, right, of that neurotransmitter. The neurotransmitter can be reuptaken by that presynaptic neuron. And that's really important because you're going to learn about specific drugs, especially one that comes right to mind, SSRIs, selective serotonin reuptake inhibitors. So this is all very important stuff. Okay, you'll see, especially in clinical medicine, you have patients who are going to be taking specific different types of medications, and you've got to be able to understand what the medication is doing there. So selective serotonin reuptake inhibitors, one class of medications you're going to learn about, they're going to not allow this reuptake to occur. Why are they not going to allow reuptake to occur? Because reuptake takes the neurotransmitter out of this synaptic cleft. We might be wanting to flood the synaptic cleft up with a certain neurotransmitter. So we'll give this class of drugs. They're SSRIs, selective serotonin reuptake inhibitors. They won't allow serotonin's reuptake to occur, because we want serotonin to be found in this cleft in higher quantities for these patients. You can have enzymatic degradation, we said. Enzymatic degradation, an enzyme can come in. Acetylcholinesterase, one enzyme you should know about. Acetylcholinesterase is an enzyme that comes in and it breaks acetylcholine down. Acetylcholine is going to be a neurotransmitter you should all know about, you should be familiar with in relation to neuromuscular junctions. And then third, we said we can have diffusion away from the synapse of the neurotransmitter. If it diffuses away from the synapse, it won't be able to then open the ion channels. Right? So if the neurotransmitter gets reuptaken, it can't open up the ion channels. If the neurotransmitter gets broken down, it can't open up the ion channels. If it diffuses away, it can't open up the ion channels. But if it makes its way out, it will open up those ion channels. So when we look at these neurotransmitters, you have to understand these neurotransmitters can be excitatory or inhibitory. Excitatory, they help generate action potentials. Okay, so we can see everything in this one picture. Next, I want to mention to you synaptic delay. Synaptic delay is getting into the time that's going to be needed for the neurotransmitter to actually be released, and then for it to diffuse across the synapse, and then bind to the receptor. This can take anywhere from 0.3 to 5 milliseconds. Synaptic delay is the rate-limiting step of neural transmission. It's the rate-limiting step of neural transmission.
Neurons communicate across junctions called synapses. When an action potential arrives at the end of an axon, a signal is transmitted across a synapse to a receiving cell. Although synapses may be either chemical or electrical, we will only look at a chemical synapse in detail. How do signals travel across a chemical synapse?
There is a narrow gap, the synaptic cleft, between the synaptic terminal of ascending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fuse with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to the receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. The neurotransmitter is broken down by an enzyme, and the ion channels close. Enzymatic breakdown is one of the several mechanisms by which transmission is terminated at chemical synapses.
Now let's check out then the postsynaptic response. Now the postsynaptic response can be either excitatory or it can be inhibitory. Now when these neurotransmitters bind to the receptors, they cause gradient potentials that vary in strength. That's going to be with the amount of neurotransmitter released and also the time the neurotransmitter is going to stay in that area. Now when we go through and we see our gradient potentials we talked about before, again, can take place either on the cell body or on the dendrites, and they're going to be signals that are going to be incoming towards the cell body. So here you can see exactly that. They're short distance signals, and then they're going to help trigger our long distance signals. And we talk about long distance signals, we refer to those as our action potentials, our long distance signals. So the short distance signals help to generate the action potentials, and the action potential is going to be generated, if you remember, in the axon hillock at the trigger zone inside of there. So again, we can see here, neurotransmitters, when they bind to the receptors, this causes gradient potentials that are going to vary in strength, again, with the amount of time that the neurotransmitter is going to be in the area and also the amount of actual neurotransmitter released. Okay? Here you can continue to compare and contrast, again, action potentials and gradient potentials to one another. And then same thing we'll see here. And these gradient potentials, we'll see their whole function is to generate either excitatory postsynaptic potentials or inhibitory postsynaptic potentials. So here's another image. It depicts what's happening. You can see this ion channel opens. You have simultaneous movement of sodium and potassium, which helps to generate then, we said, gradient potentials, short depolarizations. Now here we can see if we have potassium channels open and chloride channels open, potassium is going to make its way out and chloride will make its way in, and this is going to hyperpolarize the membrane and take us away from an action potential. And here when we talk action potentials, you can see your sodium voltage gated channels open up, allows sodium to rush in during one phase of the action potential, which we refer to as depolarization, repolarization, hyperpolarization we talked about there. So here we can see we have two types of postsynaptic potentials, excitatory postsynaptic potentials or inhibitory postsynaptic potentials. So here what we'll do is we'll look at excitatory synapses and excitatory postsynaptic potentials. And then we'll talk about and we'll look at inhibitory synapses and inhibitory postsynaptic potentials. So when we talk excitatory postsynaptic potentials, I'd like you to know that they're going to function to help trigger an action potential distally. They're going to help trigger action potentials distally at the axon hillock, we said, of the postsynaptic neuron. Now, when we talk inhibitory is going to reduce a postsynaptic neuron's ability to generate an action potential. So here we can see when we're talking excitatory synapses, the neurotransmitter binding opens chemically gated channels. This allows simultaneous flow of sodium and potassium, we said, in opposite directions. The sodium influx greater than the potassium efflux is going to lead to a net depolarization. And that's called an excitatory postsynaptic potential. So sodium's influx, you'll see, is greater than potassium's efflux. And this leads to a net depolarization, which we call an excitatory postsynaptic potential. It's not an action potential. Remember, this is generating an EPSP, an excitatory postsynaptic potential. And then when we talk about excitatory postsynaptic potentials, EPSPs, what they do is they help trigger action potentials. So again, we said they're not action potentials themselves, but they're going to help trigger action potentials if that excitatory postsynaptic potential, if that EPSP is of threshold strength. Remember, we said an action potential is only going to occur if we hit threshold. So if the stimuli is strong enough to take us to threshold, then an action potential will occur. So when we talk EPSPs, they help trigger action potentials. And they can spread to the axon hillock. where then they can trigger the opening of voltage-gated channels, and this will cause the action potential to be generated. So here we can see that. Here we have our cell at a resting voltage, and then here we can see a stimulus comes in. The stimulus comes in and it's an excitatory stimulus, so it causes this local region to depolarize. However, it doesn't allow that region that depolarized to hit threshold. So we won't have an action potential generated. Right, we said we have to hit threshold. Then in inhibitory postsynaptic potentials and inhibitory synapses, they reduce the postsynaptic neuron's ability to produce an action potential. What they do is they make the membrane more permeable to potassium or chloride. If potassium channel's open, it's going to move out of the cell. And you saw that voltage is going to become even more negative because it's a positive charge leaving the negative environment. So it's going to become even more negative. Or if chloride, it's negative, makes its way in, makes the inside even more negative. Therefore, neurotransmitter hyperpolarizes the cell. So we talk inhibitory synapses. Here, the neurotransmitter is going to hyperpolarize the cells. You can see that here. We're at our resting voltage. Stimulus comes in. Stimulus causes the cell to hyperpolarize, taking us even further away from threshold. And that, we said, is not going to promote an action potential. So we have EPSPs or IPSPs, those graded potentials, can generate either excitatory postsynaptic potentials or they can generate inhibitory postsynaptic potentials. So basically saying the neurotransmitter, once it gets released from the presynaptic neuron, and then it goes and binds to the postsynaptic neuron, there you can see it can help excite it to either try to get it to generate action potential, or it can hyperpolarize it and take it even further away from threshold and not allow it to generate an action potential. All right, so let's talk neurotransmitters. I'd like you to know neurotransmitters, along with electrical signals, are the language of the nervous system. We have 50 or more neurotransmitters that have been identified. Most neurons make 2 or more neurotransmitters. And the neurons can exert several influences. And these neurotransmitters, they're usually released at different stimulation frequencies. And we'll see they're going to be classified by structure and by function. So the first neurotransmitter that I'd like you to know about is going to be acetylcholine. Acetylcholine is the first neurotransmitter identified. It functions as an excitatory or an inhibitory neurotransmitter. It is broken down by acetylcholinesterase. I had mentioned to you. It gets broken down by acetylcholinesterase. We talk acetylcholine. Acetylcholine, you can see, is going to be synthesized from acetic acid and choline. When it gets broken down to acetic acid and choline. So then it can get regenerated by this enzyme, choline acetyltransferase. Choline acetyltransferase. Acetylcholine is released at neuromuscular junctions by some ANS neurons and by some CNS neurons. There's a disease I'd like you to know about called myasthenia gravis. We talked about that a few minutes ago. In myasthenia gravis, you'll see the acetylcholine receptors are blocked by antibodies or they're destroyed. Nonetheless, the amount of receptors is decreased. So it causes then the muscles not to work. So then the acetylcholine can't exhibit its effects on the neuromuscular junction, and it leads to the muscle not working. Next then we have our biogenic amines. Now when we talk about our biogenic amines, they are a class of neurotransmitters that includes our catecholamines and our indolamines. Now when we talk about our catecholamines, this includes dopamine, norepinephrine, and epinephrine, which are synthesized from our amino acid called tyrosine. And when we talk about our indolamines will include serotonin and histamine. Serotonin and histamine, we'll see serotonin is going to be synthesized from the amino acid tryptophan. And when we talk histamine, histamine is going to be synthesized from our amino acid histidine. These neurotransmitters are broadly distributed in the brain. They play roles in emotional behaviors and our biological clock. They are released by some ANS motor neurons, especially norepinephrine. And if we have imbalances, they're associated with mental illness. So here we can see the formation of dopamine, eventually norepinephrine, and then epinephrine. We can see we start off from this amino acid tyrosine, we said. Then we have tyrosine hydroxylase, the enzyme, come into play. Along with oxygen, they form L-DOPA. L-DOPA then undergoes decarboxylation, and it releases carbon dioxide, and DOPA decarboxylase, the enzyme, converts it to dopamine. And then dopamine can get acted on by dopamine beta hydroxylase and be converted to norepinephrine. And the norepinephrine can be converted to epinephrine thanks to this enzyme PNMT, phenylethynolamine N-methyltransferase. Next, then we have our amino acids. When we talk amino acids, the main one I want you to know from there is going to be GABA. GABA is generally inhibitory. GABA is generally inhibitory. And then last, we can see we have gases and lipids, or our gasotransmitters. This will include nitric oxide, carbon monoxide, and hydrogen sulfide gases. And they're going to bind with G-protein coupled receptors in the brain. These are lipid-soluble, these gastrotransmitters. And they're synthesized in demand. Nitric oxide is involved in learning and formation of new memories. And we'll see nitric oxide helps with brain damage in patients that have had strokes. It's also involved in smooth muscle relaxation in the intestine. And we'll see hydrogen sulfide gases, they act directly on ion channels to alter function. Now, when we talk our neurotransmitters and their functions, you'll see they have a great diversity of functions. We can classify them by their effects or by their actions. When we talk about classifying them by their effects, they can be excitatory versus inhibitory. And when we talk about actions, their actions can be direct versus indirect. So here when we talk effects, excitatory versus inhibitory, neurotransmitters' effects can be excitatory, where they'll be depolarizing, or they can be inhibitory, where they're going to be hyperpolarizing the membrane. Now, the effects are determined by the receptor to which they bind. GABA and glycine, usually inhibitory. Glutamate, usually excitatory. Acetylcholine and norepinephrine, they bind to at least 2 receptor types with opposite effects. Acetylcholine and norepinephrine bind to at least 2 receptor types with opposite effects. Acetylcholine we'll see is excitatory at our neuromuscular junctions in our skeletal muscle, and acetylcholine is inhibitory in cardiac muscle. So you can see acetylcholine and norepinephrine can bind to at least 2 receptor types with opposite effects. Now, we talk direct action versus indirect action. When we talk direct action, the neurotransmitter is going to bind to and open ion channels, as we saw, and then it promotes rapid responses by altering the membrane potential. We saw that with acetylcholine, and we can see that with amino acids also. Here we can see the receptor is closed. Ions are not allowed to make their way through, but as soon as a ligand binds to the receptor opens up, allowing those ions to flow across. Now, when we talk indirect action, in indirect actions, we'll see neurotransmitters are going to act through intracellular second messengers, usually G-protein pathways. Indirect actions are broader, and they exhibit longer-lasting effects similar to hormones. This includes our biogenic amines, our neuropeptides, and our dissolved gases. So here we can see our G-protein linked receptor. Here we can check out its mechanism of action. This system is indirect. When a neurotransmitter binds to a G protein linked receptor and activates the receptor, this receptor then activates the G protein. The activated G proteins then activates adenylate cyclase. Then adenylate cyclase converts ATP to cyclic AMP. Cyclic AMP is going to be the second messenger. There are other second messengers that I'd like you to know about, and this will include cyclic GMP, cyclic GMP. Number 2, you have diacylglycerol, DAG, it's abbreviated as diacylglycerol, diacylglycerol. And third, calcium. They're all examples of second messengers, as is cyclic AMP. As is cyclic AMP. The second messengers are important because the second messengers then are able to regulate ion channels if they need, or activate enzymes, or even interact with nuclear proteins. There's another protein that I'd like you to know about. It's called guanylyl cyclase. Guanylyl cyclase. Guanylyl cyclase converts GTP to cyclic GMP. All right, so we can see here when we talk neurotransmitter receptors, we have two types, channel linked or G protein linked. All right, here we saw G protein linked. Here we saw channel linked. Next here, then we've got a couple of tables that are going to go through a few neurotransmitters, and I don't want you to know all of them. I'd like you to know acetylcholine, norepinephrine, dopamine, and serotonin. I'd like you to know about those 4 neurotransmitters. I'd like you to know about all of them, but so much we could do. All right, we saw this didn't even work. You can listen to what she says here. All right.